Hi all, Dr. Clark here again. And for forest management, this lecture we're talking about planting and the management plan. And when I say planting, we're going to talk more about what type of seeds or seedlings, what type of planting are you going to have. It's not the actual use of certain tools to plant trees. We'll get into tools of the trade. In a couple lectures down the road, we'll talk about what the different types of planting tools are, the hay bar, and those kind of things. We'll talk about those as we progress. But there are different methods to planting. Of course, you can have things like this, manually planting seedlings, where you hire a bunch of young individuals, typically young individuals, to walk across the mountainside and plant trees, sometimes randomly, other times they're walking in transects and they're planting them on a certain line. There's lots of different ways to do this. And this kind of brings me over to what we discussed last lecture, and that is the most important piece of forest management, of silviculture, is a plan. Now, you have to remember that a lot of forests, majority of the forests that we're talking about, when you're managing them, and let's say you've came through and this forest had been cut and now it's going to be replanted. Well, you replant it on this date, you're not going to be able to harvest it for 20, 30, 40, 50 years. Okay? So a management plan is very important for basically the next manager, the next person that's going to have your position, the next person that's going to make that decision on that chunk of land is going to look at your plan, how it was planted, what it was planted with, what's the spacing, if there's a certain spacing, when were you going to go back in and thin it, if you're going to do that, what was the soil like the day of planting. There's all kinds of things that are important for that plan. So individuals who might harvest it, down the road, they can understand, well, at what point can you expect to harvest this? Or what point can you expect for the forest to be back to, something similar to what it was prior to cutting? Okay, so let's look at kind of seeding or planting methods. Okay, the one that often comes up, and especially in today's society, often comes up as, why don't we just allow it to regenerate naturally? Okay, so I get this question often, and I'm sure forest managers and people who work at the Forest Service get this question often, and that is, why don't you just allow the trees to come back? Okay. Well, that's fine, and there's methods to cutting a forest to allow for that to happen. We talked about those last lecture. If you leave seed trees or if you leave shelter wood on a cut, and then you have some natural capabilities of allowing for natural regeneration. Okay. The other thing is, there are some benefits or some things that regeneration will allow for the landscape. First of all, it allows for numerous types of tree species to be established. Okay? This could be good, and this could be bad. Okay? It depends on what the purpose of the land is. If the purpose of the land is to cut the trees 30 years from the date that the tree is planted or germinated, then maybe you don't want that many separate tree species. Okay? Natural regeneration allows for dispersion of fertile seeds. And these fertile seeds are genetically different from each other. Not always, but at least there's some genetic diversity. Okay? The effectiveness of whether or not natural regeneration can take place will be determined, by these different things. Okay, how many tree species are going to be established there? Is it the right tree species? Are you going to have competition? Okay, is the fertile seeds what's the fertility rate in the seeds? Are the conditions right for the seeds to even be fertile? Okay? In some cases, the seed covering has to be, rubbed away or has to be soaked in water. It has to have a certain pH maybe of the soil in order for that to germinate. So natural regeneration is a lot of abiotic conditions have to be in place for natural regeneration to occur in a timely manner. Now, eventually, it's going to occur, most likely. Okay, and that would be things like soil moisture level, the temperature. Okay, which diseases are out there. So this is going to be biotic. What kind of predators are out there? So what's your rodent population? What's your bird population like? These can all affect natural regeneration rates. Okay. There are also other things that we have to think about. Do we really want the forest to regenerate naturally? Okay. That's a big question. Artificial regeneration is when we take seeds or seedlings and we put them on the harvest site. Okay, you either plant them, okay, or you just overlay them, so you can just seed spray them, and there's lots of ways at which this can occur, and we'll talk about a little bit of that. But how you apply the seeds is not nearly important, in my opinion, as to what kind of seeds or what kind of seedlings are you selecting. Okay, so you could select seeds, which what I mean by that is these seeds have been harvested. Okay, maybe they're harvested from mature trees, and either the seeds are kept in a sterile environment, they're cleaned so you don't produce a bunch of diseases. Okay, and that's the seed that you're going to put on the landscape. Okay, or they're grown in a greenhouse or in a closed environment where the seedlings have good soil. They can get kind of a head start. They have fertilizer, those kind of things, and the seedlings can get a head start before you put them out on the harvest site. Okay, select seeds are natural seeds. It's just the environment in which they're either germinated or the environment in which they come from may not be exactly natural. So in other words, how many seeds you're going to put on. If it's artificial regeneration, how many seeds you're going to put on per acre or whatever it is, whatever your measurement is. How many seedlings you're going to plant. Where those seedlings came from. The age of those seedlings. The spacing of those seedlings. Okay? So select seeds, are natural, native seeds. Okay? These other two are not natural seeds, or they don't occur naturally. Okay? And that would be GMO seeds, or genetically modified organisms, or genetically modified seeds or seedlings. And then a new technique, which we call CRISPR, which is also genetically modified, but in a different way. Okay? And I'm going to cover both the GMOs and CRISPR, in a couple slides, but I just want to say that there's really kind of three types of seeds that you can select. Natural ones that have been harvested from the environment, okay, where the exact species of tree, everything like that, okay, and you can apply those to the landscape. GMOs where they've been modified, genetically modified, or CRISPR where the seeds have been or seedlings have been modified with a CRISPR technique. Okay. So that management plan that forest management plan that we need to come up with, that silviculture management plan, needs to address what kind of seeds or what kind of regeneration are we going to do. Is it going to be natural regeneration? Then you don't need to talk about GMOs or CRISPR or anything like that. If you're going to select artificial, then you have a little bit more of a choice. Do we care about genetic diversity? Okay. If you care about genetic diversity and having a very diverse group of organisms, okay, maybe it's the same species, but you have a lot of genetic diversity. So if a disease comes through, then maybe some of the individuals will make it and some won't. Then you really are looking at natural seeds, these selected seeds. That's kind of the important piece. If you're not interested in genetic diversity and actually you want to limit genetic diversity, so all the trees are uniform size, uniform growth bands, uniform everything, then you probably want to select a GMO or a CRISPR seed. Disease control, okay, if you want none, you're probably going to look at natural, okay, or very little control over the diseases. And if you want a bunch, you're going to select a GMO or a CRISPR. Growth control, same thing. Natural means they just grow at whatever rate it is that seed's genetics naturally is going to allow for. GMO or CRISPR, you could control some of the growth. Okay? Other things, do you need some type of temperature tolerance? Do you need pH tolerance, moisture requirements? Is the landscape that you're growing these seeds on, is it not actually fit or good for the species of tree that you're interested in growing? Then you might need to select a GMO or CRISPR that has some of these tolerances, or you can develop one that has some tolerances to different temperatures or pH. And like I said before, seeding, how you go about seeding the landscape is, I don't want to say it's not important, because it's important in the sense that the way that you seed today determines the way that you prune or eliminate or whatnot tomorrow, and the way that you harvest 30 years from now. Okay? And I shouldn't say tomorrow, maybe 5 years from now, and then 25 years from then. Okay? So seeding will determine that. There are many different ways to seed. Again, you can seed with mechanical equipment. So if you have a way to get into the site, you can seed it just like an agricultural field would be seeded. Okay, you can have certain amount of spacing that you can seed. You could broadcast seed it, which means that you just basically shoot seeds across the landscape. Where they lie, which ones germinate, you don't know. And so that can be done by aircraft, that can be done by lots of different mechanisms. Okay. Again, how much effort do you want to go through after seeding? Okay. If you're seeding with mechanical equipment, or if you're seeding seedlings, meaning that you're hiring people to put seedlings in the ground at a certain distance away from each other, then maybe down the road you have less maintenance, you have less actual man hours going in and maintaining that forest. If you seed it by aircraft, which is quick, okay, literally takes a few minutes, seed it, okay, but then 5 years later, you might have to hire people to go in and remove trees that are too crowded so you can get as much growth from that forest as possible. Okay, so these are the different methods. Direct seeding, or how you seed, is important. Okay? But the type of seed is far more important, in my opinion. Okay? So let's look at some of that example. Natural, I think everybody is pretty familiar natural means. Natural means pollen, ova, okay, they come together, they produce via, meiosis, they produce a offspring. Okay? Or in the case of majority of plants, many offspring. And it might take many different pollens, many different ovas, many different offspring. Okay? Genetically distinct offspring. Okay? I think most people are familiar with that. Okay? GMO and CRISPR, though, there's a lot of information out there, and some of it, quite frankly, is just wrong. Okay? GMO gets a bad rap, and I'm not saying GMOs are good for everything, but they get a bad rap because people don't understand what it means to be GMO. So let's look at that. Okay? First, let's look at the costs. Okay, so if we're looking at a cost benefit, let's look at the natural benefits. So let's look at just seeding naturally. So either leaving shelter wood or seed trees, or landscape trees are going to reseed the area naturally. Of course, we talked about this a little bit before. There's no equipment to allow for a tree to do it on its own, and there's no labor. Okay? Now, I shouldn't say no, because how you cut the forest might determine the labor and things like that. It is harder to cut a forest using, maintaining shelter or seed trees. It is more difficult, okay? Because removal of the tree can be more difficult. Okay? But some of the major benefits: less equipment, less labor, so that's a lower cost. Okay, the seedlings that do develop, that are occupying the landscape, they have naturally shaped roots. Okay, so if a seedling's growing, and there's rocks there or whatnot, those roots will grow around the rocks. They'll become established. So windstorms aren't just going to blow the seedlings out. Okay? The seedlings that are put in, okay, or the seeds, well, if it's seeds and they're dropped on the landscape, they'll have natural shaped roots too. But the seedlings that are stuck into the ground, planted, they're not going to have naturally shaped roots. Their roots are going to fit whatever container they were grown in. So that could cause, and often does cause, a little bit of a lag in growth. So you plant that seedling, and often they have a lag because they have to grow their roots. They have to send out more ground roots in a given region than naturally occurring seedlings, because the naturally occurring seedlings are going to find the easiest route. The ones that are planted in the ground, they have a big ball of roots that are planted straight down, and that's not really how roots grow. They grow out mainly, especially in the trees that we harvest. Typically, they grow out, and so you have to wait for that to take place. So again, costs less. There's no site preparation, so you just allow for the seeds to come in. And then there is obviously natural pest and disease resistance, because genetically these seeds are not all the same. Okay? But what are the costs? Okay, first of all, you have to leave some trees on the landscape. So this is a cost via cutting, but it's also a cost via, you're not harvesting that wood yet. Okay? So depending on how many or what portion of the trees you're leaving on the landscape, okay, that could be a major cost in the overall net operating cost. Natural costs also, you don't have control over the insects, the birds, the rodents that may eat these seeds. Okay? You don't have any control over that. Okay? You have no control over the spacing or the number of seeds in a given area. So you can have huge clumps of seedlings, and that because you got 5 seedlings in a foot by foot spacing, only one's going to make it. So that means 4 seedlings are basically are a waste. Okay? You don't know what the competing vegetation is going to do. How many other tree species do you have? How many shrubs do you have in that region? Okay? How long is it going to take for your naturally occurring seeds to germinate and grow to a mature enough size to outcompete some of that other vegetation in the region? Well, it could take an extra 10 or 20 years, depending on the habitat site, in order for that to occur. And in fact, maybe the competing vegetation, especially if it's invasive vegetation, maybe it never happens. Maybe your seeds never get established in certain portions, and you would have to replant or wait for mature trees to again reseed the environment because this competing vegetation is just too much. Okay. The other main natural cost is that you do not get the superior growth rates that you do with artificial seeds. Okay? Artificial seeds, for the most part, have been selected for higher growth rates than natural occurring seeds. Okay? And so that component means that you got more time to harvest when seeding naturally. All right, now let's talk about GMOs, or genetically modified organisms. And I just want to lay it out there so you understand what a GMO is. Now, you might see lots of negative information on GMOs. I mean, you can go to a grocery store and you see packaging and it'll say no GMOs, like GMOs are going to kill you. Well, maybe they will, maybe they won't. I'm not here to change people's minds on whether GMOs are bad or good, but I'm here to educate you on what a GMO is, and that in fact, people that say, I'm against GMOs, well, in fact, you probably have consumed a massive amount of GMOs in your life, and daily you're probably consuming massive amounts of GMOs. And it depends on your definition. Okay? If your definition is the definition that GMO is given, then for sure, no matter who you are, you've consumed GMOs on a daily basis. Okay, so what is a GMO? It's any organism whose genetic material has been altered using genetic engineering techniques. Now, this is where the definition gets what I call shady, cloudy, not really clear. Well, what do you mean by genetic engineering techniques? So let's look at another little piece of data. So the USDA has approved genetically modified crops, and it's approved these crops. These are the only crops that have been approved for genetically modified organisms, okay? And the different number of seed types are here. Now, there have been lots of other seed types. Okay, there's about 10 crops and about 150 different seed types, okay, that have been approved by the USDA for GMOs. Okay? Now, I'm not showing you information on plants that aren't considered food, okay, or aren't considered consumable. These are only consumable plants, okay? There's 10. Okay? Non-consumables, I mean, pretty much the majority of flowers that you can think of, okay, there's probably a GMO version of it. Okay? Lots of trees. Okay? Lots of organisms out there are GMOs, but we do regulate what food varieties can be GMOs. Okay, and so there are 3 types of GMOs. Okay? Those that have been genetically engineered within the same species. Okay, so for example, let's say we take tomato, okay, and we say, all right, this is one species of tomato. Here's another species of tomato. We're going to take the genes. I'm sorry. Here's one individual of tomato. Here's another individual of a tomato. Okay? We're going to take the genes from this tomato and we're going to manipulate the genes in this tomato to make a hybrid tomato. Okay? That's GMO. Now, you can probably go to the store and you can buy packets of what we call hybrid seeds. Okay? And in fact, they might even have GMO crossed off. But under the definition, they're still GMO. It's still genetically modified, but it's within the same species. The one that most people don't like is number 2 and number 3. That's when you take a species of tomato and another species of tomato, and you take the genes from a separate species and put it in. Okay? Or maybe it's not even a tomato. Maybe you take a tomato's genetics and you're mixing it with the genetics of, let's say a fish, at least one gene from a fish, maybe a growth rate gene from a fish, and you're sticking it into a tomato. Okay? That's across species. There's also across kingdoms. Okay, that was an example across kingdoms: plant kingdom, animal kingdom. Okay, a lot of people have issue with the across species, across kingdoms, but very few people know that even within a species, if you're selecting for a single trait, that's still a genetically modified organism. Okay? And so I want to just draw individuals' attention to that, because let's look at something like this. Okay? Most of you probably have no clue what this organism is. Now, this organism is called tesonite. Okay? This organism is a grain or a grass that evolved in Mexico. Okay? And probably in North America too, until the climate changed and then it kind of receded down into Mexico. It's a warm weather, it's a warm weather plant. Tesonite, we think about maybe 7,000 years ago or so, is when Native Americans started altering tesonite to become corn. Okay, so this is corn, sort of. Okay, and it took Native Americans a very long time, okay, to select pollen from different tesonite individuals and even some different separate species of tesonite. Select the pollen, mix it together, see what you get. Okay? Eventually, this single grain on a stalk plant became multi-grained, much larger, different kind of stalk. Nothing even remotely is the same between the plants, okay? Except for some genes that carry from the ancestor to the adult. Okay? But again, this was genetically engineered. Now, was it engineered in a lab? No. Okay? So if that's your problem, if you don't like GMOs because it's engineered in a lab, that's fine. But this is still a genetically modified organism. It was engineered to be corn. And in fact, it's now went from tesonite to corn that can't reproduce on its own. Okay? So this could reproduce on its own, this could reproduce on its own, some varieties of this could, and now we're to modern corn that's grown on the landscape that really can't reproduce on its own. Okay? It needs the help of humans, most varieties. Okay? Then we've taken this and we've done other things with it. Okay? And this is where people have a problem with GMOs, is we now taken corn and we've inserted genes into it to make it so you can spray Roundup on it, right? And it doesn't destroy the corn, or it's freeze tolerant. Right? And And those alterations, majority of those alterations didn't come from the genes within the plant. It didn't come from the genes within a sister plant or, another species that's closely related. It came from outside. It came from other organisms, and we inserted those genes. So just to be clear, though, genetically modified organisms are organisms in which the genes of an individual are genetically engineered. I would argue, and I do argue, that taking pollen from one plant and giving it to another plant is still genetically engineering. Okay? It's just you don't know what you're going to get if you just transfer pollen. If you transfer genes, then you know exactly what you're going to get. So one way is very ancient, before science advances in scientific information, and the other way is modern science, where you can manipulate single genes. All right, now let's bounce over to one other new technique. Okay, and this new technique, it's great for, I guess, the agricultural corporations, agricultural businesses. It's great for scientists, many scientists. And that's CRISPR-Cas9. And I'll go through what CRISPR-Cas9 is, but I first want to start with this. In 2018, the USDA declared that CRISPR-edited foods are just as safe as ones bred the old-fashioned way. So in other words, CRISPR-edited foods are not considered GMOs, and they're not regulated by the USDA. Now here, let's jump into what CRISPR is. Okay? So CRISPR is a technique that was evolved in bacteria. Okay? So bacteria evolved a technique to basically have an immune system that remembered when they were infected by a certain strand of DNA. So if you don't know, viruses will inject their DNA into a bacteria. Okay? And then that bacteria, that DNA makes it into the bacteria's DNA, and that bacteria will replicate that virus. Okay? But bacteria have an immune system, immune response, called the CRISPR system, CRISPR-Cas9, and there's quite a few other CRISPR systems out there. Okay, Cas9's got the most information on it, but there's lots of others. Okay, but this system recognizes foreign DNA and it removes it. Okay, so what I'm showing you here is that DNA will come in. Okay, the DNA from a foreign body will come in, and CRISPR has basically these little cutting mechanisms, enzymes that will cut the DNA and remove it, and then replace it with DNA that is deemed good, I guess. Not good necessarily, but non-foreign. Okay? So that's the general mechanism. Basically, you have scissors that'll go through and it recognizes a certain region. It cuts that region out and then replaces it with new DNA to fit that region. What scientists have done is they've manipulated the system. So now we can say, hey, you know the gene that makes it so mushrooms turn brown and start to rot? Okay, you know that gene that allows for that? There is a gene that allows for that rotting mechanism. Okay, why don't we remove that gene? And why don't we replace it with a gene that doesn't do that? Or why don't we remove a small section and replace it with a gene that makes that period much more extended? Okay? So you cut it out, and then you replace it with the donor DNA. The donor DNA is whatever you want. As a research scientist, it's whatever you want. You can manipulate that DNA however you want. So you can remove things that you don't want to see. You can remove mutations that might happen in the DNA, and you can replace it with a non-mutated version. Okay? You could replace a non-mutated version with a mutated version. You can do whatever you like, and it's fairly cheap. Okay? When I say fairly cheap, we're talking you could probably manipulate a single gene in a plant to give, some mechanism that you want, maybe resistance to a certain blight or something like that. You can manipulate it. Now, I'm not talking about doing all of the tests and things like that to ensure that it works, but you can manipulate for probably $50,000 probably. That's dirt cheap. Roundup ready corn, Roundup ready soybeans, things like that were created in a different mechanism, the GMO mechanism. Okay? We're talking millions upon millions of dollars before you could get a Roundup ready corn or soybean, and years and years of research. This can be done much quicker and much more precisely. Okay? So, what does that mean? Well, it means that CRISPR-Cas9 organisms are not going to be regulated by USDA according to their 2018 ruling. And it means that companies are starting to create things that have genetically modified regions, GMO, okay, but using this system. There's nothing saying that you cannot manipulate the DNA of this organism, add any donor DNA you want. You can make your own donor DNA, and that's exactly what scientists are doing. They're not stealing it from other species. They're making their own donor DNA based on other species. So they might have a gene from another species, and they know the adenine, thymine, guanine, cytosine repeat, what it is, how big that gene is, and then they just slap it in, and now you have that protein being created inside whatever organism you're interested in. Okay? So there are probably a lot of people that after they find out what CRISPR-Cas9 can do, they're going to boycott CRISPR-Cas9 foods. Okay? But currently under USDA, no one has to tell you. They don't have to tell you that your food has been genetically modified. If it was CRISPR that did it, okay? Because they've determined that it's as safe as naturally occurring food. Okay? So let's look at some of these crop technique modification techniques. One that most people don't have a problem with, and guaranteed that you've consumed food, even if you are against GMOs, you consume food that has this happen to, and that's crossbreeding. You find two separate varieties, in some cases, two separate species. You mix their pollen, and you see what you get. Okay? This is how we got apples. This is how we took crab apples and made modern apples. Honeycrisp, whatever the apple is that you're interested in, Fuji. All of these were made in that manner. You mix pollen, you produce a fruit, you see what you get. Does it taste good? If it does, great. Now we just clone it. Okay? We don't take the seeds from that because the seeds mean it's genetically modified. Now we just clone the tree that grew that fruit. Okay? That's crossbreeding techniques. There's lots of them. That's how a lot of seedling trees are developed currently, is through crossbreeding. Because the tree that's going to grow fastest, etc. You have certain traits that you want. You don't want the genetic diversity. You want uniform tree growth. Okay? Mutagenesis. Okay? This is creating mutants. Okay? So you can introduce radioactive material that mutates the DNA, and then you test it out and you see what kind of characteristics it gives you. Okay? We've been doing this for a very long time. Okay? And that's how you get basically ruby red grapefruit. Okay? Instead of being pink and it's red, that's a mutation. Okay? That was introduced through mutagenesis. Polyploid. Okay? So instead of being diploid or tetraploid or something along those lines, you create a ploidism by removing or adding chromosomes. Okay? And you manipulate the chromosomes, which manipulates something to make it, in many cases, non-fertile. Seedless grapes, seedless watermelon, these kind of things, they are a manipulation of polyploidism. Okay? Still, genetically modified, all of these. Protoplast fusion, this is when you're transferring cells and you're fusing cells together. Okay? Often by doing that, you can make sterile individuals. This is great if you have a specific trait that you don't want to breed out of your system. So use a protoplast fusion. Okay? Transgenesis, this is the one that most people have a huge problem with. And that is when you're inserting genes from a new organism, a different organism, whatever it might be, to create a different variety or something with a desired trait. Okay? So that's transgenesis. Okay? And then genome editing, we just talked about that. CRISPR-Cas9 is a genome editing tool. Again, the nice thing about that scissors and tape system is what you tape in there is determined by the individual doing the taping. Okay? So can you combine gene editing with transgenesis? Absolutely. And it's done often. Could you just look up what gene code you need and create that code artificially and then insert it? Absolutely. It's been done. People are doing it right now. Okay. So what I really want to just kind of clear up, just to make sure people understand, crop modification techniques, these are all technically genetically modified organisms. They would not occur in a natural way. You would not occur polyploid, seedless watermelon, seedless grapes. You wouldn't occur mutagens, maybe crossbreeding, but highly unlikely because most of the time these are transferred from individuals that would never even occur in the same region, not alone would even bloom at the same time. So the pollen ova of these guys are normally not ever going to come into contact with each other. Okay? Protoplast never would happen. Transgenesis, genome editing, not going to happen. Okay? So next time you look at GMOs, okay, think about what is going on. Is your issue, if you have an issue with GMOs, which I do not, okay, I'm just letting that be out there right now. I do not have an issue with GMOs. Okay? But if you do, is your issue with transgenesis? Or is your issue with everything? If your issue is with everything, the food types and the food varieties that you are going to be able to consume, pretty much zero. None. Okay? No fruit, no vegetable. And if you don't believe me, just look up, okay, broccoli. Do you know that broccoli is mustard? It's a mustard, okay? So we took a mustard, we made broccoli, cauliflower, Brussels sprouts, cabbage. They're all the same species, just manipulated just a little bit. Okay? And so if you go to the store and you buy a head of broccoli and it says no GMOs, just realize, yeah, that organism is genetically modified, because without modern science, modern agriculture, and the manipulation of crossbreeding, you never would have broccoli. Okay? It is manipulated mustard plant. Okay? All right. Now, why choose GMOs or CRISPR? Well, here's a pretty good example of what's going on. So, if we look at pasture land or secondary habitat. And we look at these habitats here and we say, okay, what is the germination rate? So we look at germination rates and we say, okay, germination rate in pasture lands or secondary lands, okay, it's somewhere a little bit below 50%. In plantations, where you're typically, when you're talking about plantation, you're selecting for seeds that have been genetically modified. Okay? And soon you'll be selecting for seeds that have had CRISPR techniques used on them. You're looking at closer to a 50% germination rate. Okay? So a little bit higher. That's not the biggest deal. The biggest deal is when we start looking at survival rates. Okay? When we look at survival rates in pastured regions or secondary habitat regions, these regions, it varies. Sometimes they're genetically modified seeds that are being put on that landscape. Sometimes it's germination by or establishment by seed trees or shelter wood. Okay, but nonetheless, you can look. There's a huge drop off. Okay, surviving year 2, you're only looking at about 20% of the trees in these 2 regions are surviving to year 2. When you look at that plantation, it's close to double. It's almost 40%. Okay? So this might be the reason why, hey, look, if we're going to have a plantation style, we should be selecting for seeds that have been genetically modified to resist fungal growth, or genetically modified to resist some type of disease, and has an extended growth rate, or can withstand desiccation for a longer period of time, or higher temperatures, higher soils, or higher pHs or lower pHs, whatever it might be. That's one of the reasons to do it. Your drop off from germination to 2 years, surviving 2 years, it's much less. Maybe 10% drop off. That's a huge deal when you're talking about planning for the future. If you're only losing 10% between germination and 2 years, after the 2-year mark, most trees aren't dying. Okay, they make it to 2 years, then they're established pretty well, unless obviously a fire or something breaks out. But they're established pretty well by the time they make 2 years. Okay. All right. So with all that, next time we're going to talk more about equipment and some technique stuff, and then we'll just go from there. So